Hey everyone, we're going to take a look at Chapter 3, start this slideshow, the innate immune system. The innate immune system we're going to take a look at. This is just a quick roadmap of where we're going. We're going to look at the external and internal defense systems. Those we know as the first and second lines of defense. This slide we've seen before, this table, and it breaks down the immune system of the host or vertebrate host into the innate immune system or the non-specific versus the adaptive, which is the specific immune system. The innate immune system that we're going to look today includes the physical barriers, and the book we're going to also call it the external defense system. So those are the physical barriers. The internal defense system are kind of the second line of defense. Those are chemicals and some cellular components that we're going to take a look at today. This is all familiar with us so far, so we're still good, right? Good. We've seen this slide a number of times. I know that I've told you this is one I really like. I think it breaks down the immune system in a very clear way. The immune response, we're going to look at the first line of defense, which is the external, and we're going to look at the second line of defense, which is the internal. We're ignoring the acquired immunity today. Innate and natural immunity really is just talking about the ability of the individual to resist infection by the means of normally present bodily functions. You know, the innate immune system provides really a broad protective defense against infection to the host. It's present before any exposure to the pathogen has ever taken place. As we've talked about before, you could have the introduction of E. coli or Pseudomonas, and just the recognition of being foreign is going to trigger phagocytic activity that will kill that pathogen, for example. The skin is non-specific. It doesn't care what it is. It's going to try and keep everything out of the body. So it's a very non-specific immune response. The innate or natural immune systems are active right at birth. And as I said, we know that it involves non-specific responses to pathogen. The innate immune system is comprised of two systems that work together to promote phagocytosis. We're going to look at the external defense system, which are really the anatomical barriers that are designed to keep microorganisms from ever entering the body. Keep them out, they can't do any damage. But if a pathogen happens to break through the host's external defenses, it falls on the internal defense system as the next line of defense. This includes some cellular responses of phagocytic cells to recognize very specific molecular components of pathogens, and it could generate either a cellular or a soluble factor response. These two components of the innate immune system can be thought really as sort of the first responders because they react very quickly to the onslaught of infectious agents, and they're really quite effective. As a rule, the innate immune system is so efficient that truthfully most pathogens are destroyed before they ever even encounter any of the cells of the adaptive immune system, and that's a good thing. First, we're going to take a look at the external defense system, which are the physical barriers of the immune system. We're going to look at the structural components of the external defense system. We're going to look at things like unbroken skin and hair. We're going to look at chemicals, including lactic acid, fatty acid, sourcin from epithelial cells, sweat, stomach acid. Then we're going to look at some organ systems and some other things like normal flora. If you look at the picture to the right, this is a great illustration of some of the external barriers. So we're going to look at chemicals and other secretions in tears. For example, lysosomes in tears can flush away microbes and also have an enzymatic reaction trying to digest and kill microbial pathogens. For example, we're going to talk about the removal of particles by cilia or the stomach acid of pH. All of those are external barriers, and if you can take a look at that, that's a great little chart for you to look at. First defense system we're going to look at under the external defense system is the skin. We know this is a really important component of our external defense system. The skin is structurally a very tightly packed set of epithelial cells that are coated with a chemical called keratin. The skin is basically impermeable to most infectious agents based on the tight packing of the cells. The keratin itself, we're finding probably just in the last 10 years or so, at least that's when I first became aware of it, that the keratin really acts as a structural support for skin cells, and without it, the cells really start to become much more fragile, and the effectiveness of skin as a defense mechanism becomes defective and less protective for the host. The dermis that we see here below in this illustration to the right, just below the epidermis, is connective tissue with blood vessels, hair follicles, sebaceous glands, sweat glands, and white blood cells included. The white blood cells that we're going to see here are what? They're essentially macrophages as a rule. Some of the secretions and protections of the skin are included in this slide. We see that sweat secreted creates a very high salt condition and contains lactic acid that helps maintain a more acidic pH. So between the salt and an acidic pH, organisms really don't like it, and it's what we term an inhospitable environment. The oil layer and the fatty acids that we see here from sebaceous glands, as well as proteins, which include lysosomes, and an acid pH also inhibits the growth of microorganisms. Next, the psoriasin is a small protein that's produced inside of the skin cells that has another antibacterial effect against microbes. We probably should put in here, and I didn't, I will try to remember to do that in the future, that one of the other components of this layer of skin really is the phagocytic cells that are contained in the dermis. These cells include, any of the phagocytic cells that we find in tissues, macrophages, dendritic cells, and mast cells. And then we can talk about normal flora as a mechanism for defense and protection, in that normal flora that's established as a colony, really prohibits the colonization by a new microbe, by a pathogenic microbe. And it does that really by competing for, the natural resources of the host, the space, the nutritional support. And so if you can keep a nice, healthy, normal flora, then you've got half a chance that microbes are not going to establish a colony there. In addition to the discussed skin that we just saw as a structural defense, other structural defenses include things like organ systems. These provide a support system for the host in a number of different ways. If we look at the respiratory system as a defense mechanism, we can see in this illustration of the lining of the respiratory system, we see epithelial cells that are ciliated, so we know that cilia is just the little hairs, and the cilia are covered by a mucous membrane. It's this mucous membrane that protects you, so that when you breathe in, if a particle of some kind or a pathogen is inhaled, it gets stuck in the mucus, and what it ends up doing is it blocks the pathogen from adhering to these epithelial cells. If it adheres to the epithelial cells, it can cause damage and can enter into the host through that route. So if you have this mucous membrane, what happens then? The cilia, as you remember from Bio 1, get irritated and they sweep the mucus into the throat to either cough out or to swallow. A couple of the other organ systems that we've discussed before and will continue to discuss because they are great external defenses are the urogenital tract and sort of the, in a continuation of looking at the processes that we just talked about with the respiratory tract, we can also see how the stomach acids are going to be involved. So if we talk first about the urogenital tract, we talked about this last week, that some of these actions are designed to keep microbes from colonizing. And so what happens? So the first thing we do is we create a really acidic pH. We see lactic acid, for example, creates a really acidic pH, and that's that inhospitable environment for pathogens. It's not inhospitable for things that normally live there, but for pathogens that aren't used to it, that acidic pH is not a great place to grow and to try and set up shop. We also talked about the flushing action of urine, that really keeping things moving, keeping the water flowing, and that flushing out of microbial organisms out of the host helps again from keeping those microbes from colonizing. And then we talked about the normal flora. We also talk about that as the microbiota. It's really just we're talking about the normal flora, the normal colonization of the host with normal flora. That normal flora helps keep the pathogens at bay, really again by competition, competing for nutrition, nutritional support, space, and other requirements needed for growth. What do the stomach acids do for us? Well, it's an incredibly acidic, very, very low pH area. So pH is between 1 and 2 because of the hydrochloric acid of the stomach. That's a strong prohibitive agent in discouraging microorganisms to grow there. So that when we talk about the respiratory system, how the cilia sweeps the mucus up into the throat to either cough or swallow. If you swallow it, those particles and pathogens go into the stomach where they encounter this incredibly low pH that's really going to prohibit them from growing. It may not always kill them, but it's not going to allow them to grow very effectively. So the question of the day, if our GI tract is filled with all sorts of normal flora like E. coli, why don't we just get sick from that? We get sick from ingesting E. coli, from undercooked meat, or even from things like romaine. But why don't we get sick if our GI tract is filled with normal flora? We'll talk about that on Thursday when we get together. The next, that was the first line of defense, so that's really the mechanical side of the chemical, the pH balance kind of a thing, and then reflexes like coughing, sneezing, flushing of urine. Second line of defense, still non-specific. It's going to include some cellular components, some soluble factors like acute phase reactants, responses like fever and inflammation. We're going to talk about all those things. And we're going to ignore, of course, acquired immunity until another week from now, or another day from now. So the second line of defense, this is that second line of defense of the internal defense of the innate immune system, or the non-specific immune system. What do we know about this? The second line of defense includes both cells and soluble factors, soluble meaning dissolved, that are involved in that second line of defense. Uniquely, or comparatively to the adaptive system, it's still nonspecific because the cells of this line of defense, the cell of the internal immune system, the innate immune system, are designed not so much to recognize a specific microorganism, but to recognize some molecules on things that are infectious in nature. So it doesn't rely on having previously been exposed to something in order for it to respond. Let's say, for an example, there's an example down below. White blood cells, phagocytic cells, can recognize a carbohydrate called mannose. It's found on the bacterial cell wall. And when it's found by a white blood cell, it's tagged for phagocytosis and death. Honestly, it doesn't matter what kind of microbe has mannose on it. It's just the recognition of mannose that targets the microbe for destruction. It doesn't have to be a specific species of microbe or or a virus or something, like you would see in the adaptive immune system. It's really just recognizing a chemical component, so it's very nonspecific in nature. Phagocytic cells, we know, are kind of indiscriminate in their digestion of pathogens. Anything it recognizes as foreign is subject to destruction by a phagocytic cell. The good news, though, is that the internal defense system tries to give the phagocytic cells a little bit of a boost. They give them the presence of some soluble factors called acute phase reactants, can assist in the process of phagocytosis and other immune responses. So that's one of the soluble factors that we're going to see involved. But there are other things that are aiding the phagocytic process. And we're going to talk about something called pathogen recognition receptors. PRRs for short. PRRs are receptors on phagocytic cells that help identify pathogens. And once identified as pathogenic, the phagocytic cells destroy them. So, for example, in the example we see above where the white cells recognize mannose, there will be a pathogen recognition receptor that will locate mannose on a microbial cell wall and target that microbe for destruction. It's the interaction between the mannose and very specifically the pathogen recognition receptor, looking for that. So this is where we're headed. So first, let's take a quick look, though, at the cells of the line of defense, the second line of defense. And we're going to spend just a few minutes on them, and then we're going to move along quickly. When we first looked at the cells of the nonspecific immune response, we looked at them in terms of where they were found. So we looked at the cells that were found primarily in the vasculature and those of the tissue. So we looked at these phagocytic cells that were found in the vasculature. We looked at, this is found in the vascular platelets, are not white blood cells, but we're going to talk a teeny little bit about them today. And then we talked about some of the tissue cells. Very specifically, the tissue cells were mast cells, dendritic cells, and macrophages. Remember, these are the really fantastic antigen presenters. They were all antigen presenters if they're phagocytic in nature, but the dendritic cells were the star pupils. And what's the role of the phagocytic cell really is to engulf and destroy something that's pathogenic. That's the issue, and that's what we're looking to do with phagocytic cells. Mediator cells are really sort of helper cells. We're going to see, for example, the basophils can release granules that will induce an immune response. And then under other, it's true macrophages and dendritic cells are also phagocytic. They're tissue phagocytes, but dendritic cells, for example, are really, as I said, really heavily involved in phagocytizing antigens and presentation to T cells, as are macrophages. Those are two strong APR presenters. Not APR, APC, antigen presenting cells. Too many acronyms for the day. We'll start with A. Where are we headed with this? So when we're looking at the internal defense system, the next thing we're going to look at are some of those non-cellular components. So we just looked at some of the cells of the internal defense system, and what their general roles are. Now let's look at some of the soluble components and take a look at those. We'll see down here, natural killer cells are clearly not a soluble component, and maybe it should have gone in another place. I don't know, but this is what we're looking. This is our roadmap for the end of the chapter related to the internal defense system. Let's call it that way. We're going to look at pathogen recognition receptors, soluble proteins, inflammation and fever, phagocytosis, and natural killer cells. Probably the most difficult concept for the day are these pathogen recognition receptors. As I said earlier, phagocytes contain receptors, and we call these PRRs, pathogen recognition receptors, that are really designed to recognize a molecule unique to a pathogen, or unique to pathogens, not necessarily one pathogen, but not found on self. So they're pathogenic, the molecules on pathogens, and the phagocytic cells have receptors on the cell surface that will discover these and will tag that organism for phagocytosis and death. We use these in some respects. We're going to see later on in the chapter how these pathogen receptors are able to help the phagocytic cells decide if something is self or non-self. And obviously we do want to have a halting system that will not have us have our phagocytes killing all of our own cells indiscriminately. So it's important that the immune system has a mechanism to identify that. So the non-self cells or non-self organisms are identified as such by pathogen recognition receptors on phagocytic cells. And I'm going to apologize in advance if I stumble over my words here, because it's sometimes I speak too quickly. I'm trying not to. These pathogen recognition receptors, a great way to think of them is that they're sensors of pathogens. They sense what's a pathogen and what's self. They're genetically coded by the host's own DNA to recognize non-self, to recognize that extracellular infectious agent. It's a genetic coding. And PRRs recognize only molecules that we call PAMPs, P-A-M-P-S, or pathogen-associated molecular patterns. I can see a typo there. That should be patterns, not patterns. I thought I corrected that. PAMPs are recognized by PRRs, and these PAMPs are found only on microbe organisms of some kind. And they do, and over time, through natural selection, I'm sure, and all sorts of genetic coding, self does not have any pathogen-associated molecular patterns that are going to be identified by phagocytes and targeted for destruction. So PAMPs we only find on the microorganisms that we want to target for destruction. I gave the example earlier of the mannose. Mannose is a PAMP, and that's what triggers that kind of microbe for destruction by a white blood cell. Are we together so far? PRRs are on the phagocyte, PAMPs are on the microbe, and PAMPs are just a molecular pattern or structure on the microbe that identifies it as foreign. So, the pathogen recognition receptors are found on macrophages, dendritic cells, and to a lesser degree these other cells, although neutrophils are actually still fairly well populated with pathogen recognition receptors, but macrophages and dendritic cells have a higher degree of PRRs on them. Once the PRR has detected and recognized something as a pathogen, what happens? What's the mechanism that allows us to destroy that pathogen? So if we look up top, we imagine up above here, we have the pathogen recognition receptor has identified a PAMP over here on a microbe. So that's happened. And it's detected it as a pathogen. So they bind off together, it detects it as a pathogen. The PRR and the PAMP bind together. This is what I said about, I have to slow down because I'm going to say the wrong word all the time. Let me start. The phagocytic cell containing the PRR detects a PAMP, a molecule on a microbe. It detects it. The phagocytic cell then binds to the pathogen, and it's that binding that activates the phagocytic process afterwards. Phagocytes are then activated. It's going to trigger the steps of phagocytosis, which include engulfing and destroying the pathogen, and simultaneously secreting cytokines, which, remember what those are? Chemical messengers, and some subsets of cytokines called chemokines. Those are the chemotactic ones, the ones that call other cells to the area. So while phagocytes are engulfing and destroying a pathogen, it's going to also start to secrete other chemical messengers saying, hey guys, I found something foreign over here, something that shouldn't be here. I want to call in reinforcements, and I want to look for all of the pathogens here, and we need to destroy all of the pathogens. So that's the purpose of these cytokines and the chemokines. The chemical messengers, the cytokines, also have other functions. The chemokines are the ones that involve movement and calling cells to the area, but other cytokines can do other things. They can increase capillary permeability. Why? Because we want those white blood cells that are kind of skulking around the vasculature to be able to enter the tissues if needed. So it increases the capillary permeability. The cytokines trigger some chemotaxis. Remember that it's going to call additional white blood cells to the area. So if we imagine that, we have the vasculature and we have pathogen having been detected by a phagocytic cell, and there are white cells, floating, stuck to the vasculature over here, the phagocytic cell releases chemotactic cytokines. And remember, these white blood cells kind of follow the breadcrumb trail from very low concentrations of chemokine, and the closer and closer that it gets to the pathogen, the concentration of the chemotactic chemical, that cytokine, gets stronger and stronger. The concentration gets stronger and stronger, and it will, at some point, because we've increased the capillary permeability, the white blood cell can leave the vasculature and go and assist in the defense of the host against the microbes that were found there. It's also going to trigger, those cytokines can also trigger the adaptive immune system. Let's see. Yeah, I think that's all I want to say about it on this particular slide. So we've established the fact that the immune system uses these pathogen recognition receptors to identify pathogens. And it does that by identifying that unique molecular structure that's only on things that are foreign, only on foreign pathogens, non-self. And it's those PAMPs that the pathogen recognition receptors will find. Let's look at some examples of a human pathogen recognition receptor. There are several different kinds. The ones that I tend to think of most commonly are the ones we call toll-like receptors. It's a strange name, but it is what it is. I'm going to refer you guys to page 34. Yep, 34 of your book as we do this. So I'll wait 2 seconds, and while I'm speaking, you can go to page 34. And we're going to talk about toll-like receptors. Toll-like receptors are simply a type of pathogen recognition receptor on phagocytic cells. So, what are toll-like receptors? Toll-like receptors, let's start with what's in a name. Toll-like receptors, toll-like. What that's referring to, and honestly I think they made this more difficult than it needed to be. A protein that's named toll was originally found in a fruit fly in the species Drosophila. And so that protein called toll plays an important role in the antifungal immunity of the fruit fly. When they were looking for an analogous protein in humans, they found one. And so they named it a toll-like receptor. So it's not exactly analogous to the antifungal activity of the fruit fly, but it was close enough for them. So they called it a toll-like receptor. What are toll-like receptors? They're glycoproteins that bind to those PAMPs, those substances of infectious agents that we talked about, those PAMPs. So toll-like receptors are just a kind of pathogen recognition receptor. What do? We know what they do. We just saw what they did. They activate phagocytosis. They release cytokines, etc., right? So we're going to do all those things to enhance phagocytosis. And it's because that phagocyte has recognized that pathogen as non-self and it needs to be destroyed. This process can destroy most pathogens that humans get exposed to before the disease is allowed to set in, before the microbe is allowed to kind of colonize the human host. And I asked you to look at table 3-1 of the book because we're going to take a look at some of the toll-like receptors. As I said, they initially found the first toll-like receptor that was sort of analogous to what they saw in the fruit fly, and they named it a toll-like receptor. Since then, they found more. And they just started numbering them. So we have toll-like receptor number 1, 2, 3, all the way up to 10. So there's 10 different toll-like receptors. And if we look in chapter 3, page 34, table 3-1, we're going to see on the far left-hand side of that table, there's a list of 10 different toll-like receptors. Toll-like receptors 1 through 6 are found on cell surfaces, and toll-like receptors, actually, no, 1, 2, 4, 5, and 6, all but 3 are found on cell surfaces. Toll-like receptors 3, 7, 8, 9, and 10 are found internally. They're found in the cytoplasm, in the endosomal compartments of the cytoplasm. So they're found inside of cells. Why do we need internal and external toll-like receptors? Well, because we know that some of our pathogenic organisms live outside of the cells, and some of them live and replicate inside of cells, like a virus. So we need some of our pathogen identifying substances inside of the cells to find those viral components, right? So despite the fact that they have similar names of toll-like receptor 1, 2, 3, whatever, each toll-like receptor identifies a different kind of a pattern. For example, if we look again at the book, toll-like receptor 1, which is found on the cell surface, recognizes the molecule of lipopeptides. And we know that those are found on mycobacteria, mycobacterium. So that's the target organism type that we see affected by toll-like receptor 1. Similarly, toll-like receptor 5 recognizes flagellin on an organism, and that recognizes and destroys bacteria that have flagella. So toll-like receptors are looking for some kind of a unique molecule to non-self, to those pathogenic organisms. And so if you look at this table, there's all different things that are going to be found on the surfaces of cells that can be identified by toll-like receptors specifically. There are going to be other toll-like receptors that we're going to see as we go through this. But for now, we're concentrating on these examples of pathogen recognition receptors. Let's see. The greatest concentration of toll-like receptors are on monocytes, macrophages, and again, as I said, neutrophils, dendritic cells, and I think that's all I care to say about this slide right now. Here's an illustration of what we just talked about regarding toll-like receptors. If we look at this one, we said there were 10 different toll-like receptors. There were 10 different types of toll-like receptors found in humans. I told you that some are found on the cell surfaces, and those are toll-like receptors 1, 2, 4, 5, and 6. And some are found in the cytoplasm. Here's the ones that are found on the cell surface, and here's the ones that are found in the cytoplasm. So imagine this is the microbe. This blue of this illustration right here is all reflecting the microbe that's there. I'm sorry. I apologize. Let's step back. The blue is reflecting the phagocytic cell. I apologize. So the blue is the phagocytic cell, and so the toll-like receptors, and they're not going to be all on the same cell, but there are going to be toll-like receptors on the surface of the phagocyte and some inside. Toll-like receptor number 1, we decided, was looking for lipopeptides of mycobacteria. And so that we would imagine is going to be an extracellular one. Mycobacteria is a gram-positive bacteria that lives outside of cells. So we're going to need a pathogen recognition receptor, like toll receptor 1, outside of the cell to find the mycobacteria that's out there. Similarly, we may need some that are inside the cytoplasm. Toll receptor 3 is inside the endosomal membrane, the endosomal compartment of the phagocytic cell, the monas, the dendritic cells, the neutrophils. They live inside. They're inside. They don't live inside. They're a structure inside of the cell. The toll-like receptor 3 is looking for viral dsRNA. And so viruses are replicating and synthesizing inside of cells. And so the toll-like receptor, or the pathogen receptor, looking to see if it's foreign, is going to be inside. Just the importance of extracellular and intracellular pathogen recognition receptors make sense to everyone. If it doesn't, make a little note to yourself and ask me a question or post me a question, and I will talk to you about it. Give another example where toll-like receptor 2 is an external one, and it recognizes the tectoic acid of some gram-positive bacteria. So that's just another example. Toll-like receptor 5, we talked about, remember this is the one that looks for the flagella, so it's going to detect motile bacteria. So those are toll-like receptors, and remember, toll-like receptors are what? They're examples of pathogen recognition receptors found on phagocytic cells. Very simple when you break it down. Almost very simple when you break it down. So wouldn't it be lovely if the only pathogen recognition receptors we had to worry about were toll-like receptors? But heavens no, it's not. There are more. There are, in fact, other pathogen recognition receptors that we need to look at. One is called a C-type lectin receptor, or CLR. We find this on monocytes, and this is the one that binds to, oh no, this is not. This is one that binds to mannan and beta-glucans that are found in fungal cell walls. And this is going to activate certain cytokines and chemokine productions that are going to result in the phagocytosis of those fungal cells. The retinoic acid inducible gene 1-like receptor, or the RLR, retinoic acid inducible gene I-like receptor, is one that recognizes RNA from RNA viruses, and it too will induce cytokines and interferons. Interferons are a kind of cytokine that inhibits viral replication. So when you see something that's viral, frequently you'll see interferon is a cytokine that's used to interrupt the replication. The action of those cytokines, and specifically interferons, ends up resulting in if you interfere with viral replication, then the infected cells are going to die, and what do we call that? We call that apoptosis. That's we're going to look at cell death as a result of those interferons and cytokines. And then another kind of pathogen recognition receptor that we're going to look at is the nucleotide binding oligomerization domain. Let's call it the NOD receptor. The NOD receptor binds a peptidoglycan in bacterial cell walls. And we see this as associated with Crohn's disease if there's a defect in the NOD's receptor. So we end up with kind of an inflamed GI tract, and we see this in patients with Crohn's disease. So in addition to the toll-like receptors 1 through 10, here are some of the other important pathogen recognition receptors that we're going to see. So that's the end of the pathogen recognition receptor discussion, but we're not really done yet with the internal defense system, right? So that was pathogen recognition is part of the cellular, it's part of the phagocytic process, it's part of the cellular components of that second line of defense. But there's more, right? We know that there were serum proteins that are involved in that internal defense system, or the second line of defense. And if you go back to that chart, you'll see on the second line of defense there are going to be serum proteins. And the soluble components are going to be what we talk about next. acute phase reactants are soluble tissue and serum substances that will help suppress or kill microorganisms. They are varied in nature, and I think, again, if we look at our book, table 3-2, did I put the chart in here? No, I didn't, but I did go through each of the acute phase reactants. So you can follow along with page 35, table 3-2, the characteristics of acute phase reactants. These acute phase reactants have different mechanisms for participating in the internal defense system. So let's go through them one by one. We're going to look at the general characteristics, and then we're going to look at some of the specifics of them. The characteristics of acute phase reactants include the fact that it's normally found in serum. We see these generally rapidly rise during infection, injury, trauma, tissue damage, all of those things. Remember, phagocytic cells don't just respond to infection, they kind of clean up cells too. So if you have tissue injury or trauma, tissue damage of some kind, phagocytic cells also go there, kind of clean up the mess, mop up the mess. And just like with an infection, cytokines are produced by phagocytic cells, and the cytokines will induce these reactions of acute phase reactants. So acute phase reactants normally found in serum increase rapidly in some kind of tissue damage, whether it's by infection, injury, trauma, any other kind of tissue damage, like a heart attack. Some of the functions include facilitating binding and adherence between microbes and phagocytic cells. Remember, we talked about how acute phase reactants enhance the phagocytic process. Some of them actually do mop up and recycle some important proteins and chemicals within the host after phagocytosis. Some are transporters. There are many, many different roles of acute phase reactants that we're going to talk about. And they're primarily produced in hepatocytes or liver cells. I did make a table. Good, I forgot. Let's look at the first one. Is the most important for us to talk about today. I'm not going to say it's the most important one. It's a very important one called C-reactive protein. And C-reactive protein is normally a trace constituent in serum. It's in very low concentrations normally, but it plays a really important role in the immune response, and its concentration will increase 1,000-fold after an infection or injury. It's a high molecular weight structure made of 5 identical subunits, and so it's called one of the pentraxins of a kind of a structure. The structure is called a pentraxin because there's 5 identical subunits, and it's very large. As I said, it's roughly 118 to 140 something Daltons in size, so it's big. It has a couple of different functions. It binds to a chemical called phosphocholine in microbial membrane and other proteins. It can act in a number of different ways. It can act as an opsonin, and if you look at that table, you'll see it can act as an opsonin, coating a pathogen, making it more susceptible to phagocytosis, and it can also activate another part of the immune system that we're not going to talk about today, called complement. It's quite a system, and we're not going to talk about it today. It would take another hour. C-reactive protein has a short half-life, about 18 hours, and it peaks within about 48 hours of response. So has a really rapid rise and a really rapid return to normal once the condition subsides. It's a really effective monitor for markers of injury and inflammation because it rapidly returns to normal after the condition is gone. So if you have an ongoing form of tissue damage, then the C-reactive protein levels remain pretty high in the serum. But if it peaks and returns, you know, after 48 hours, and then another 18 or 20 hours after that, when we start to see it going down, then the tissue damage has subsided. So we use C-reactive protein as a marker for healing, and for kind of acute types of inflammation. In the lab, where do we see it? We see C-reactive protein as a really useful marker in diagnosing myocardial infarction, tissue damage from myocardial infarction. So we're going to see it used in that respect in the laboratory. How else do C-reactive proteins how else are C-reactive proteins binding as a receptor? C-reactive protein can also bind to, as I said, some of the other components, chemical constituents of bacteria and fungi and parasites, like peptidoglycan, the ribonucleoproteins and phospholipids of bacteria, funguses, and parasites. And by binding to those chemical constituents, it makes those microbes more susceptible to phagocytosis. So C-reactive protein is a really important and acute phase reactant in monitoring the condition, but also really making the microbe more susceptible to phagocytosis and is marked for phagocytosis. As I said, C-reactive protein is capable of opsonization. That's just coating a microbe to make it more susceptible to phagocytosis. It's capable of glutenating and precipitating and activating something called complement. It can agglutinate cells and platelets. As I said, it can bind to receptors on some phagocytes to promote phagocytosis after it's after microbes were detected. And as I just mentioned in pretty fair detail, that it's a great predictor of stroke and heart attack, and tracking of tissue damage when you see an increased level of C-reactive protein in the serum. So when you think of C-reactive protein, think of opsonization, and opsonization, complement activation, which we'll talk about another time, but also think about it related to being increased in heart attacks and strokes, someplace where there's a strong degree of tissue damage. The next acute phase reactant is serum amyloid A. It's a major protein secreted during the acute phase of inflammation, that we're going to talk about briefly. It also increases 1,000-fold, but it's a low molecular weight compound. Maybe I think it's around 11,000 Daltons, if I'm not mistaken in my memory. Yeah, it's not in the book right there, but I'm pretty sure I'm right. Serum amyloid A has a couple of different roles. It's kind of a cholesterol cleanup. Serum amyloid A has a really high affinity for cholesterol. It's attracted to it. So it removes cholesterol from cholesterol-filled macrophages. Now remember, macrophages are what? They're a cleanup crew, right? So they're cleaning up loose cholesterol, and then the macrophages need to be cleaned up, and that's done by serum amyloid A. and so it cleans it up at a site of injury. So that's one effect. Serum amyloid A also recruits immune system to sites of inflammation, and it's thought to play a role in cholesterol metabolism. Serum amyloid A has a stronger response in bacterial infections more than viral, and its other primary function, really, that I don't even really see here, is that it is an activator of macrophages and monocytes. So it's an effective activator of the phagocytes in primarily bacterial infections. Alpha-1 antitrypsin, despite what it says, is not just against trypsin, and we're going to talk about that in a second. Alpha-1 antitrypsin is a serum protein that increases during acute inflammation. It protects the tissues from enzymes of inflammatory cells, white blood cells, and it's protectively effective against an enzyme called elastase, as well as trypsin, but it's elastase is one of the ones that we really think about it when we talk about alpha-1 antitrypsin. And so the way it works is alpha-1 antitrypsin is an inhibitor of these enzymes, of these proteases, these enzymatic, these enzymes against proteins. So the protective nature of alpha-1 antitrypsin shows that it's a protective model. So if you don't have enough alpha-1 antitrypsin, and the lungs, for example, don't have enough alpha-1 antitrypsin, this enzyme elastase is free to destroy lung tissue, and as a result, the lung cell loses some of its capacity to expand and contract, that elasticity, which leads to severe lung disease like emphysema. Sometimes makes breathing really, really difficult. Alpha-1 antitrypsin also has a function as a kind of a cleanup protein as well. It counteracts some of the proteolytic enzymes, the proteases that get released by white blood cells during inflammation. So it is it's not just protective, but it also kind of cleans up. It's kind of a cleanup crew as well. And so if you look in the book, its primary function is as a protease inhibitor, but remember, it can also kind of clean up the release of the proteases from white blood cells. Fibrinogen, we're going to see that, you'll see this a lot in other classes as well. Fibrinogen is an acute phase reactant protein that's involved in clotting. We call it a clotting factor, and it's required to form a clot. So what happens, you get damage, whether it's a cut or a crushing injury, you get damage to some vasculature, and we have to form a plug in that hole in the vasculature to prevent microorganisms from entering the body that way and entering into the blood system. So fibrinogen is one of the factors that's important to create a clot formation, and in fact it gets activated and it forms kind of a mesh that will cover over the hole in the vasculature. Fibrinogen levels are going to increase with tissue inflammation or destruction. So if I said, whether it's a puncture wound or it's a crushing wound, fibrinogen is going to come and try to repair the damage with the rest of the clot formation processes of coagulation, but it also plays a role in when there's tissue inflammation. Fibrinogen is also thought to play a key role in the inflammatory result in developing of rheumatoid arthritis, and is found in contributing to some coronary artery disease in women. And the way this happens is that fibrinogen, the presence of an increased presence of fibrinogen in the bloodstream makes the blood really pretty viscous. And as a result, it's sort of it slows things down and it promotes clotting or aggregation of red blood cells and platelets. Aggregation just meaning clumping of red blood cells and platelets within the vasculature. That can cause some damage over time, and you end up with, potential for coronary artery disease. But if we look at the primary function in table 3-2, what is it? It's involved in clot formation. The other things are kind of secondary actions of fibrinogen as an acute phase reactant. Haptoglobin, we'll speak quickly about. It's a large alpha-2 globulin. What is its purpose? It binds irreversibly to free hemoglobin, and it protects the kidneys from damage. So it additionally prevents loss of iron by excreting free hemoglobin out in urine. Your body is, pretty protective of the things that it synthesizes, and hemoglobin, when it's synthesized, it requires an awful lot to do it. It requires iron. It requires a significant number of nutrients and things that are ingested or manufactured, and so it doesn't like to really give it up by peeing it out through the urinary system. So once the hemoglobin is bound to the haptoglobin, the hemoglobin complex, instead of being peed out, gets removed and filtered by the Kupffer and parenchymal cells of the liver and by the spleen, and it gets reused. Haptoglobin is one of those things that's a monitoring acute phase reactant as well. We can see it in the lab. And if we see a very low haptoglobin level, if they suspect hemolysis, they may do a haptoglobin level. And if it's very low, it's low because it's all bound to free hemoglobin. So you don't have free haptoglobin. So that's kind of a monitoring one as well. But again, the function is primarily to bind hemoglobin. Ceruloplasmin, another large acute phase reactant, whose principal job is to transport copper proteins. It's principally designed for copper transport. It's a copper transporting protein found in the plasma. I shouldn't have tried to read it. I should have just said it. About 70% of all the copper in the human is bound by ceruloplasmin. It also plays a role in iron metabolism and some other immune responses like histamine regulation. Ceruloplasmin stimulates the immune system to fight infections, repair injured tissues, and promote healing. But in its functionality as a transporter of copper and binding of copper, if you have a depletion of ceruloplasmin, you're going to find that in a disease called Wilson's disease. This causes the body to absorb and retain excessive amounts of copper, and the copper then deposits in tissues like the liver and brain and kidneys, eyes, and causes very severe tissue damage, necrosis, scarring, and all of the organs become much less functional, resulting in things like liver failure, damage to the central nervous system, the brain, and the spinal cord, and there are very, very serious effects from this disorder. How can it be treated? Well, you can chelate copper if you know that you need to. The copper can be chelated out of the blood system, out of the host. But you can also give a liver transplant. Liver transplants sometimes are needed as a result of liver failure. So that's ceruloplasmin, and again, primary function binding copper and oxidizing iron. Two main functions. So copper transporting and oxidizing iron during iron metabolism. Coming close, complement. We're going to spend very little time on this today because it's a very, very complex process, and we have, I think, a whole chapter on it later on. Complement is a series of at least 20 serum proteins. They're enzymes that it's like rolling downhill. It's involved in the mediation of inflammation, but it also is involved in opsonization, chemotaxis, cell lysis, and it involves the antibody-antigen reaction to stimulate white blood cells in those reactions. So complement, the process of complement involves multiple pathways triggered by, among other things, in some cases microbes and cell walls of microbes, that results in cell death. So it's a pretty active and effective acute phase reactant in acting as an opsonization and a lytic factor. So the role of complement is ultimately to kill a microbe. Let's talk briefly about the process of inflammation, which is another one of those secondary lines of defense of the immune system, of the immune system. This is within the second line of defense further. And what is inflammation? It's really the body's overall reaction to injury or invasion by an infectious agent. Inflammation dilates the blood vessels, increasing the blood supply to an area, and it's going to cause things like redness or erythema, swelling, edema, heat, or pain. Erythema and edema should be in red. Those are two words you really need to know. What are the mechanisms of inflammation? Well, at the site of infection, the inflammatory response causes macrophages and mast cells to release, those cytokines, those specifically chemokines that not only result in vasodilation, but also triggers the white blood cells to come to the site of infection. So we know that chemokines do that. But we also see that it causes vasodilation, widening, right, of the vasculature. The white cells come to the site from capillaries in the surrounding area and tissues. The chemokine as I said, we've talked about before, just as a definition, the chemokine is the chemotactic kind of an agent, right? It's a type of cytokine. So just want to make sure we remember that what we're talking about. What else happens to this? As the white blood cells are, we'll talk in a minute about how the chemokines get the cells there. What else happens with inflammation? There can be selectins that bind to circulating lymphocytes and cause lymphocytes to move along the wall. I think actually that should be leukocytes. So instead of this saying circulating lymphs, I think that's a typo and it should say leukocytes, causing leukocytes to move along the cell wall. When we talked the other day, we talked about how white blood cells, leukocytes, I swear I changed this. I think I might have, an uncorrected PowerPoint up here. So I'll make sure to put the right one up on the portal. But that should say leukocytes. So as remember, white blood cells can be living in the circulating pool, the vascular pool. And if we think of something like neutrophils specifically, they're in the circulating pool only, a few hours, right? 6-8 hours, I think. And then they sort of skulk along the vasculature under the action of selectins. And as they skulk along the vasculature in the marginal pool, skulk, that's the technical term, through the marginal tool, the release of other surface selectins can cause the white cells to just move along that cell wall, skulk along the cell wall. And when a chemokine calls it, the white cells can then bind to the endothelial cells of the cell walls and crawl between them in the mechanism called diapedesis. So what happens in inflammation is really that those cytokines start to call the white blood cells to move into the tissues. And it requires that process to take place. It requires selectins to bind to the cells of the circulating pool. That causes them to bind off to the endothelial cell walls of the vasculature, and then they crawl out between the endothelial cells in a mechanism called diapedesis. Chemotactic agents then lead the white blood cells to the site of infection. Remember, from low concentration of chemokines to highest concentration of chemokines. And that process of movement towards the site of infection or towards the site of damage is called chemotaxis. Here's a picture of that. If we look at this, and it's if we start at the top, we see some form of this is within. Let's see, you can't see it here, but at the site of infection, macrophages and mast cells release chemokines that cause vasodilation. So it widens the vasculature. Selectins are released and they bind circulating leukocytes. Yeah, see, I was right to say leukocytes, not lymphocytes. They bind circulating leukocytes to the vasculature. So here's the binding of the circulating leukocytes from the circulating pool to the marginal pool. And I joke, they skulk along. Once they bind, they skulk along this endothelium until they are, some conformational changes take place, and it allows the deformability of a leukocyte to squeeze between the cells of the vasculature out into the tissues. And then they'll follow from a very low concentration of chemokines to a higher, ever increasing level of chemokines to the site of infection by the mechanism we call chemotaxis. So this mechanism of moving along, binding, and moving along the endothelial cells and squeezing through is diapedesis, and then the movement towards the site of infection by chemical stimulus called site of chemokine is called chemotaxis. One of the other main purposes of the secondary line of infection, or secondary line of defense, is phagocytosis. This is a big one. The main purpose of the inflammatory response that happens during the second line of defense is to attract cells to the site of infection or the site of damage, and to remove the damaged the damaged tissue or the pathogens from that area. And how does it do it? It's done by phagocytosis. So we know that phagocytic cells are primarily these ones. These are the most phagocytic cells, although we know there are others. Neutrophils, oh, my book just fell over. Neutrophils, monocytes, macrophages, and dendritic cells are all phagocytes, along with others, but these are the most phagocytic cell ones. And this process of phagocytosis can be enhanced by some of those APRs, the acute phase reactants that we just spoke about. So let's look at this process and see how the process of phagocytosis works. we see that there is a pathogen outside of the cell, and the pathogen has recognized it as something that's foreign, or it has been, this could be a particle that needed to be destroyed. But in this case, this is a pathogen. And so we see that the microorganism is maybe has a coating on it, maybe it's opsonized, maybe it is however it happens, so an APR could opsonize it. But in any event, the phagocytic cell binds to the microorganism and will start to engulf it. So it starts to bring its cytoplasm up and around the microbe until the engulfment by the cytoplasm fully surrounds the microorganism, and that formation, once that microorganism is totally inside of the phagocytic cell, we call this area, the little component containing the microbe, a phagosome. That's this area that's totally surrounding. Oops, there it goes. This is a phagosome. It's the little compartment containing the microorganism. What happens next is that some granules are released and enter into the phagosome as well. Lysosomal granules they're released and they contact the phagosome and they fuse in with the phagosome, forming what's called a phagosome. So here's the original organism with the lytic agents in it, and that's called a phagosome. The digestion of the organism starts to take place once it's in contact with the granule, the contents of the granules, the lytic agents, starts to digest it and break it down into really small little pieces. And we know what happens then. Those little small pieces they're not in here, but they bind to the MHC, and the MHC leaves the endoplasmic reticulum up to the Golgi apparatus and up and out in a process called exocytosis out of the cell. So this is the mechanism of phagocytosis that we're going to see. I think we'll talk about it again in just a second. What are the monocytic phagocytic cells? And this is a great picture. This is there's the antigen, and it's being brought into the phagocytic cell. It's being engulfed by the phagocytic cell. So what kinds of mononuclear phagocytic cells are we talking about? We talk about monocytes and macrophages. And they're found, monocytes are when they're found in circulating blood. Macrophages are when they're found in tissues of the body. These are really effective phagocytic cells. What do we know about them? They arise from the bone marrow. They can divide in the tissue. And their goal really is to ingest and destroy material like bacteria, kind of debris, damaged host cells, tumor cells. They stay in the peripheral blood for a couple of days, about 70 hours, and then migrate to the tissue where they get very large, doubling in size, and they are then called macrophages or tissue macrophages. If you remember when we talked about macrophages, we talked about how they're named according to the tissue location. Liver cells are called Kupffer cells. Macrophages found in the brain are called microglial cells. And then phagocytosis is known to take place in greater degree in the tissues. There's a high concentration of phagocytic cells there, so it's a great place for phagocytosis to take place. So this again is the mechanism of phagocytosis in a little more, just a slightly different picture. I think that is very, very similar to what I just went over from the picture that was in the book. This one, I liked this picture, and I liked this just to go through it one more time. So The phases of phagocytosis after damage to the tissue by either trauma or the result of microbial multiplication. It's initiated, phagocytosis is initiated. The initiation results in chemotaxis, the chemical calling for cells to come to the area, the attraction of white blood cells and other cells by chemicals. Once that happens and cells come to the area, the phases of phagocytosis are as follows. The first phase is called adherence, and that's that physical contact between the phagocyte and the microorganism. Sometimes acute phase reactants can opsonize the microbe or other particle to make it really susceptible to phagocytosis. But that first physical contact between the phagocyte and the microorganism is called adherence. And so we're going to see that first. That's step one. And once it adheres to the phagocyte, it starts to be engulfed into the cytoplasm by the phagocyte. The phagocyte starts to close its cytoplasm around, and it's going to enclose the microbe in a small vacuole or a small component called, and we're going to term that a phagosome. They also, in this picture, called it a phagocytic vesicle, but we like to think of it as a phagosome. The formation of a phagosome is when it's completely surrounded by the cell membrane. Then lysosomal granules from within the phagocyte, the phagocytic cell, come in contact with, here's the phagosome, lysosomes of the phagocyte come in contact with a phagosome containing at that point just the microbe, and they fuse together. Lysosomal granules then release their contents into the phagosome, and once that happens, once those contents are released and they're bound together, it's called a phagosome. So here you can see the lysosome is fusing with the phagosome. It becomes, and all the contents are released into them. It's called a phagosome. And here it is where this is starting to digest the microbe, as we saw just a few minutes in the other slide. Digestion by enzymatic methods happens and destroys the microorganisms inside. A number of different killing mechanisms can take place to kill the microbe inside of there. Inside of the vacuole. One might be that lytic process, the enzymatic process that we just talked about, but another one involves the formation of hydrogen peroxide inside that phagosome, that when the phagosome is formed, there is a very quick and rapid burst of oxidative energy that's formed, and it happens when the process in ingesting and creating that phagosome takes place. Once that happens, that oxidative burst actually binds, the oxygen and binds off to some hydrogen, and hydrogen peroxide is formed within the vacuole. That combined with the intracellular enzyme is really rapidly lethal to the microbe that's there. So there's a couple of different methods of killing. The first was the lytic process. The second is the oxidative burst resulting from the use of oxygen within the vacuole and combining with the hydrogen later. After the material inside the phagosome is digested, there can be a little bit of undigestible material left, and what happens to that? It gets released from the cell via exocytosis. The digested pieces of the phagosome of the microbe in here also can be broken down, and the microbe can be broken down and be presented as with the MHC as an antigen presenting cell. At this point, we're just looking at phagocytosis and seeing it exit the cell through exocytosis. Opsonized microbes, actually, if we just go back here for a second, this process is actually really pretty quick. If a microbe is opsonized, it can be ingested by a phagocyte in about 20 seconds. And killing is very, very rapid, almost immediate. So the process of phagocytosis is quite complex, but it's actually really speedy and effective. This just brings both of those pictures together for us. We're going to look at putting white cell migration and phagocytosis together in one picture, so we can get a really quick overview of phagocytosis and diapedesis together. So imagine there's some form of tissue injury here. In this case, we see a puncture by a pin, puncture the skin surface and introduce some bacteria to the area. This releases cytokines or chemical signals, things like histamines, that result in dilation of the vasculature, so the vasculature gets bigger. There's increased blood flow, there's heat, there's pain, those kind of hallmark signals of inflammation, swelling. And as the excess fluid from the widening of the vasculature comes, the excess fluid leaves the blood vessel and goes out into the tissues, and that's where you get that swelling in the tissues. The cytokines in the first step also can signal white cells to come. Those are the chemotactic ones. And we know that some of the chemical signals do a couple of things. Some of them call white blood cells to the area. Some of them also, the selectins, do what? They cause the white blood cells that were in the circulating pool to bind to the endothelial cells of the vasculature, and they move along the vasculature, along the endothelial lining of the vasculature to the site of infection. And once they're there and they've gotten to the site of infection by chemotactic migration, the white blood cell squeezes through the endothelial layer through a process we call diapedesis. They squeeze through and they move further into the site of infection. So you can see, oh, look how red and swelled that looks. It's, after that's had that tissue injury. That's the diapedesis. The phagocytes and the macrophages then consume the bacteria, clean up the debris, clean up the stuff that was left behind, and the tissue eventually heals and the inflammation goes down. And our last step of the innate immune system, the natural killer cells. Natural killer cells are the first line of defense we have against cells that are virally infected, cells that are infected with intracellular pathogens like malaria, for example, and even tumor cells. Natural killer cells can recognize any damaged cell and eliminate it without prior exposure to it, giving the immune system time to then activate the adaptive immune response to specific T and B cells. Oops, back, yep, back, forward. Okay. Let's just go back for one more second to this. No, okay. So when we're talking about the action of natural killer cells in the innate immune response, we think of this as the cellular defense. Natural killer cells get activated by exposure to cytokines, which are increased in viral infections, for example. And once they're activated, natural killer cells produce additional cytokines and things called colony stimulating factors, which we'll talk about another week when we're talking about cytokines in more detail. But they help recruit other T cells to the area. This process of activation of natural killer cells influences, it's one of those things that crosses both the innate and the adaptive immune response, in that the natural killer cells can immediately kill a cell, kill a pathogen, or a cell containing a pathogen, for example, a cell containing a virus. And it's going to bridge that gap towards adaptive immunity by calling additional T cells into the area to be exposed to the pathogen and begin the process of either cytotoxic T cells or T helper cells, or even in some cases it may be B cell activation, but less so. I think we'll really talk more about cytotoxic and T helper cells. So if we look at this figure, and I believe this is in the book in small pieces. This is a small piece of the figure 3-7 that's in the book. If we look at this, we're going to see figures 3-7 illustrates the actions of natural killer cells. So in this example, we're looking where natural killer cells are they're constant surveyors, because remember, they're a non-specific cell. They're always looking for antigen presentation cells. And so when they encounter an antigen presenting cell, an APC, they look and see, they have a receptor that looks to see if there's an MHC on the surface of the antigen presenting cell. If the MHC has, an antigenic protein, remember that would be found right here in this peptide cleft that we talked about for chapter 2. If there's a little antigen in the peptide cleft there, then killing is going to occur. But if there's no antigen in the peptide cleft or no protein in that cleft, the natural killer cell recognizes that there's nothing foreign attached to this target cell, attached to this antigen presenting cell, and as a result, it sends inhibitory signals back to the natural killer cell, and the natural killer cell will have no killing happen. So if you look at this illustration, there's the natural killer cell. It identifies, it finds the MHC on the surface of an antigen presenting cell. The recognition occurs, between the natural killer cell receptor and the MHC, and it notices that there's no protein and no antigenic material there. And so it signals this process, signals natural killer cell to, X, no killing, there's nothing to see here, this is self. So this APC is sort of looking at self, and so it's recognizing self-tolerance here. Remember, if there had been something foreign inside the target cell, it would have been brought to the surface, but APCs also monitor for self, and when they do, there's no polypeptide, there's no peptide, antigenic peptide brought to the surface in the cleft. So recognition of an MHC without an antigen attached to it, an antigenic peptide, results in no killing by the natural killer cell, and that's a good thing, right? The other two that we can look at, the other two processes of natural killer cells, is that if the natural killer cells encounters an infected cell, for example, we see in the top illustration here, a virally infected APC, a virally infected target cell. So if the natural killer cell encounters an infected cell, and the antigenic markers get expressed on the cell surface of the APC, which we see here. These are antigens on the surface of the APC. The natural killer cell can actually bind to it. It has a receptor to bind to those antigenic markers on the cell surface. And when it does, um, it releases those granules we talked about yesterday. It releases perforins and granzymes, perforins and granzymes, to kill the infected cell by a process we call apoptosis. That's cell death. So when a natural killer cell has a receptor that recognizes an antigen on the surface or a marker on the surface, it targets that cell for cell death. And finally, if the natural killer cell encounters a antibody that has been bound to an antigen on an antigen presenting on an APC and antigen presenting cell. What happens here? So we have the antigen presenting cell, and it brings the antigen up to the surface, or there's surface antigen. There's antigens on the surface of this cell that bind to a very specific antibody. The antibody binds to the antigen on the surface of this cell, and it's stuck. So the antibody is stuck to that. What happens is the natural killer cell has a receptor for antibodies that are bound to cells, and when it does, if it finds that reaction, that receptor finds the antibody that's attached to a cell. Again, enzymes are released. There's no inhibition. Enzymes are released. Those same kind of lytic enzymes are released. They enter into the cell and they kill the cell by natural means, and we call that cell death apoptosis. So those are kind of the three actions of natural killer cells that we are concerned with. The activation, it's no killing, it's self-tolerance, it is identification of virally infected cells or cells that have antibody coating the cell, and it's usually going to coat some kind of something that has a surface antigen. So those are the three things we're looking at. But when it finds something that's infected the cell, whether it's virally infected or it's identified by antibody coating it, apoptosis is the result of both of those because those are infected cells. Yay, that's the end of the chapter. We can look at the summary in your spare time. I'm not going to go through it. We just went through it. Please let me know if you have any questions at all. There's our references. If you have any questions at all, let me know. We will talk about anything you have questions about on Thursday, and I will see you then. Thanks. Bye-bye.